Watch a cellular colony in its prime. One cell divides into 2, 4, 8. It is an engine designed to multiply exponentially. In industrial biomanufacturing, it's intuitive to look at this aggressive multiplication and assume a direct correlation. Growing a larger physical mass of bacteria will automatically yield a larger volume of your target product. We scale this biological process up into massive steel vats to produce vital compounds, most notably antibiotics like penicillin. We fill a tank with nutrients, seed it with microbes, and let the organism grow. Yet, when you measure the actual chemical outputs inside those tanks, the data completely contradicts that linear assumption. You can cultivate a massive volume of cellular material and end up with almost none of the antibiotic you set out to produce. Mastering industrial yield requires us to look past the sheer volume of cell division. We have to map out the strict kinetic limits governing how these biological systems actually consume and convert matter. This chart graphs a batch culture of penicillin. Initially, the biological fuel, lactose, rapidly drops while cellular biomass surges. Growth then hits an apex, and the biomass curve flattens. The paradox: our target, penicillin, begins its steepest upward trajectory only after the organism stops growing. Engineers refer to this as kinetic decoupling. The timeline for primary biomass formation is entirely separate from the timeline for secondary product synthesis. To maximize the output of the factory, you cannot simply let the organism grow unchecked. You must precisely manipulate the chemical environment at the elemental level to force the cells into the correct metabolic state. Before we can fix the output of a macroscopic steel bioreactor, we have to deconstruct the exact microscopic fuel requirements driving a single cell. We have to scale down. A cell is essentially an intricate chemical refinery, and it requires highly specific inputs to build its physical architecture. It needs large quantities of macronutrients, elements like carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur, physically linked together to construct carbohydrates, lipids, and proteins. To process those building blocks, the cell also requires micronutrients, trace elements like iron, copper, and zinc. These lock in as cofactors, activating the enzymes that make the cell's machinery run. This documentation reveals the extreme speed of that machinery. The generation time, the time it takes for a microbial population to double, can be as short as 2 to 20 minutes. Apply a 20-minute generation time to a closed volume of liquid. Billions of rapidly dividing cells simultaneously pull carbon and nitrogen from the suspension, violently stripping the environment of available structural components. An unmanaged, closed biological system is literally programmed to consume itself into starvation. Returning to the batch culture timeline, we can observe the exact moment that environmental collapse occurs. Look at the substrate lines. The curves for lactose and ammonia scrape the very bottom of the chart. The fuel is gone. Biology responds to this crash with a metabolic shift. The organism detects that the primary nutrients have vanished, so it halts cellular division and triggers emergency survival mechanisms. It begins synthesizing secondary metabolites. Compounds like penicillin are not used for structural growth. They are chemical weapons excreted exclusively during this high-stress phase to kill off competing microbes and secure whatever scarce resources remain in the fluid. This creates a severe catch-22 for bioengineers. You require vast amounts of cellular biomass to manufacture the product at an industrial scale. However, the biological trigger to actually synthesize that product only activates when the mass is starving and its growth has stopped. The engineering mandate becomes clear. How do you artificially sustain a biological system in a highly productive state of starvation without letting the cells actually cross the threshold into death? The engineering solution is to completely abandon passive batch processing and move to an active, continuous feed system. The graph on the right details the kinetics of this managed continuous culture. Notice the stepped line labeled sugar feeding. Engineers pulse exactly enough substrate to keep the existing biomass alive, intentionally depriving the culture of the surplus required to resume division. Delivering that precision requires constant data. You cannot see the exact metabolic state of billions of microscopic organisms inside a sealed steel vat. Instead, the system relies on gas exchange as a biological proxy. Watch the curves for dissolved O2 dipping and CO2 formation rising in direct response to the feeding steps. Oxygen uptake and carbon dioxide exhaust provide instant, real-time telemetry on the metabolic speed of the entire culture. By locking the sugar feed algorithmically to the rate of gas exchange, the system prevents the violent substrate crash of the earlier model. Monitoring exhaust gases allows engineers to permanently suspend the culture in its highly productive secondary metabolite phase. Placing the isolated penicillin curves from both models side by side reveals the ultimate impact on yield. In the unmanaged batch culture, the peak is sharp but short-lived. The total product yield is constrained by the natural life cycle of the closed environment. In the actively managed continuous system, the plateau of production is extended indefinitely. The total area of yield massively expands. This difference is not a biological accident. It is the direct mathematical result of overriding the organism's natural limits through active kinetic management. Optimal industrial yield is achieved by strictly aligning environmental inputs to match the precise metabolic outputs of the cell. The principles that solved the penicillin paradox quickly scaled beyond the manufacturing of early antibiotics. This exact kinetic logic governs the processing of lactose by lactic acid bacteria, underpinning the global dairy and food infrastructure. It applies to the industrial scaling of yeast fermentation, determining the yields of both advanced biofuels and international beverage production. Regardless of the specific microbe you are cultivating or the target molecule you are extracting, the engineering mandate is identical. Modern industrial biomanufacturing relies heavily on the integration of precise sensor data to perfectly balance biological processes against strict chemical limits. By accurately predicting generation times and mapping nutrient feeds against real-time gas exchange, engineers learn to control the microscopic environment. Decoding the kinetic limitations of a single, invisible cell gave humanity the blueprint to construct the macroscopic factories that synthesize the modern world.